Welcome, everybody, and thank you for joining us. My name is Caroline Hernandez. I am here on behalf of the Rights of Ways Habitat Working Group, which is hosted by the University of Illinois, Chicago. This is the second of the Research Roundtable Energy, Transportation, and Pollinator Nexus webinar series. And we're really trying to hit that sweet spot between where research meets application. Want to take a second to thank our co-hosts and the organizations that have helped us put this together, including Southern Company. We wouldn't be able to do it without you all, and it's been, just really a pleasure working with you on this so far. So we're looking forward to the next couple as well. Okay. Before we get started, I am going to take a second and let everybody introduce themselves. At UIC, we also have Iris Caldwell and Claudia Kuklinska, who are helping me on the back end. So thank you both for keeping this running smoothly, and to the participants, if you receive a message from either of them, I would definitely encourage you to pay close attention. Claire, do you want to introduce yourself?
Sure, thank you, Caroline. My name is Claire Ike. I work for Southern Company in their Environmental Affairs organization and focus on wildlife and rights-of-way vegetation management issues, including pollinators. We're really excited to be part of this webinar series and are excited about the presentations today. Thank you.
And I'll introduce myself. My name is Ashley Bennett. I work with the Electric Power Research Institute, and at EPRI I focus on vegetation management research, mostly on rights-of-way, but on other utility lands. And today I'm looking forward to telling you about 3 different right-of-way research projects that we have underway.
Great, thank you. So reason we're here today, I'm excited to introduce you all to the Rights of Way and Pollinator Conservation webinar. Really excited about this webinar. It's going to fit in nicely with the first of the series, which was Pollinator Habitat Establishment and First Year Management. And if you missed that webinar, would encourage you to go to the Rights of Ways Habitat Working Group webinars page to go and watch it, which is also where you will be able to find a recording of this webinar. And this is part of a 4-part series, as I mentioned in the past. So this is the second one. And this webinar series, this Research Roundtable webinar series, was designed to really imitate something that the Rights of Ways Habitat Working Group had done in the past, and we called it a science roundtable. It was at one of our in-person meetings, and similar to what we're doing with this series, we had researchers present, and then we got the practitioners and researchers together at a table to discuss the findings, to talk about ongoing research and research needs. So that is the goal of what we are doing today and for the rest of this series. Is we are planning on highlighting current research that's ongoing and that's relevant to rights of way and vegetation management. We're hoping to facilitate some really good discussions about related research, either past research that has happened, current and ongoing research that we might not have gotten to in this series, and potentially future research. Finally, we are looking to all of you to help us identify any kind of research needs, gaps that could benefit pollinator habitat creation and any organizations that are looking for something specific. So within those conversations after these presentations and the discussions, keep an eye out for any kind of research needs that are brought up. Finally, we're hoping that this is going to be your opportunity for collaboration. We're hoping to introduce you to new people and organizations that you might not have met before. And that's where you would discuss those research needs. Are there opportunities to collaborate on these research needs, and what can this mean for the future? So with that, I'm going to turn it over to Claire for some housekeeping items.
Thank you. Yes, we do have a few housekeeping items we want to remind everyone of. Please remember to keep yourself muted and off video except for during the breakout discussions. We're also asking everyone to update your Zoom name to include your organization. Given the number of people on this call, this really helps for all participants to know who's on the call without kind of going through a formal introductory process with everyone. To do that, you can click on the 3 dots in your video box and select rename, and then you can just type in the name of your organization following your name. Also, if you're having any technical issues, please contact Claudia Kuklinska via the chat box. All other questions, of course, are welcome in the chat box that will be visible by everyone. And finally, as Caroline mentioned, we are recording these presentations and we'll share with the group afterwards. Next slide, please. For today's agenda, the first half is going to be focused on 3 research lightning presentations, and the second half will consist of a breakout session portion for about 40 minutes, and then everyone will be brought back into the full Zoom meeting and have a larger group discussion on highlights and themes from the breakouts. For our speakers today, we have 3 exciting talks planned. Dr. Laura Russo will be talking about how vegetation management can promote pollinator diversity on rights-of-way. Ashley Bennett will be speaking about how the utility industry is using IVM to support pollinators on rights-of-way. And Dr. Emilie Snell-Rood will be speaking about the nutrition of roadside plants for pollinators and implications for managing roadside habitat. Next slide, please. And first, we will have Dr. Laura Russo. Laura, whenever you're ready, and Caroline, once you're passed the baton, the floor is yours.
Thank you. Let's see if I can. There we go. Thank you for coming to this session today. I'm going to talk about a research project that I helped on in Pennsylvania on a power line right-of-way, where we were trying to look at different vegetation management strategies and to compare them for how they promoted pollinator diversity. And by pollinator here, I'm just referring to flower visiting insects. So I did this work while I was a postdoctoral researcher at Pennsylvania State University. I'm currently an assistant professor at the University of Tennessee in Knoxville. So I just wanted to start by acknowledging my project partners on this. So the lead was Dr. Mahan, who's at the Penn State Altoona. And then I had co-authors Hannah Stout, Dana Roberts, and Bradley Ross. And then we had a lot of collaborators that were energy companies and people who managed the power line rights of way. And these collaborators helped with funding and access to the sites. We also got identification assistance from Sam Droege with the USGS and Andy Dean at Penn State University. And there are, because this is a long-term research power line right of way, there are a lot of publications associated with the site, not necessarily just the pollinators, but with the site. So if you're interested in learning more about the research site, you can go to one of these websites and it will have, it'll provide PDFs of the publications related to the site. So just for context, the site was located in central Pennsylvania, not far from the Penn State University. And it's a power line right of way, and it's known as State Game Lands 33. It's a research area that has been studied continuously since 1953. And the idea of this area is that it is integrated vegetation management. So the objective is to create stable, early successional plant communities that don't interfere with the power lines, ideally. And then because of those vegetation management schemes, there are a lot of research questions that follow in terms of how does that vegetation management affect wildlife, for example. It was started as a demonstration area because hunters in the region were concerned about the use of herbicides in managing the vegetation and whether or not that would affect deer. And then the intent was originally to show that selective herbicide use doesn't cause damage to wildlife or the value of the land, and both in terms of game and non-game wildlife. I also have a little inset here showing the research plots at the Powerline right-of-way. So the plots are these little squares, and you can see that the full distance from the one plot to the last is about 2.7 kilometers. And there were two high volume foliar sites, two low volume foliar sites, one low volume basal site, and one site that was just cut by hand. We also conducted a pollinator bowl survey, and it was placed in the middle of these 6 research sites. So this is just for context later when I'm talking about the different treatments and how they compare to each other, so you can compare them visually, spatially. So the vegetation management scheme, the concept is that there is a wire zone in the center, which is 90 feet wide, and then there's a border zone on either side of the wire zone that's 50 feet wide. And so it's possible to have slightly larger shrubby vegetation in the border zone, but the wire zone has to be cut very short. And so the management of both the wire zone and the border zone can be slightly different, and they might have slightly different plant communities. And the treatments we were looking at, the hand cutting, which involves no herbicide use, so that's just mowing or cutting by hand with clippers. And then there are the 3 other treatments are combining both mechanical and herbicidal treatments, and these are used by the utilities. So the low volume foliar, which we're calling LVF, you can see here in the table, there were 2 different low volume foliar plots. And we're just comparing the liters applied per hectare in 2016 for reference, because they're treated roughly every 4 or 5 years. And then the herbicides used for that are listed here as well, if you want to compare the specific herbicides that are used in the different treatments. And then the high volume foliar, you can compare the liters per hectare. It's pretty different between the 2 different plots. So there's a categorical effect of the vegetation treatment, and then there's also a continuous effect of liters per hectare usage. And then the low volume basal application, which uses similar liters per hectare as the low volume foliar, but different herbicides, and is applied differently. So the foliar is obviously applied to the foliage, and the basal is applied to cut stems for shrubby vegetation. There's a lot of other research that's been conducted at this site. So because it's been studied intensively for wildlife since 1987, there are publications about bird diversity and abundance, mammals, reptiles, amphibians, butterflies, and plants. And all of those are available on the websites I showed earlier. And there's also a long-term study of the Powerline right-of-way as early successional habitat for breeding birds that prefer that early successional habitat. Our objective in coming to the site was to extend the research to pollinating and flower visiting insects. So we had a few different ways of sampling pollinators. Our primary method was a hand net. So I've included a photo of Brad with a hand net here sampling the Powerline. And I just wanted to include the relative sampling effort for bees and non-bee insects in the 2 different sampling methods. So for nets, the net samples, we had about 2,300 bee specimens and 744 non-bee specimens, and 126 bee species and 179 morphospecies of non-bee insects. The pollinator sampling, we did the bee sampling for 2 years and the non-bee sampling for 1 year. So the total sampling effort by net was 192 hours of sampling. And then we also supplemented this with one bowl survey, bee bowl survey. And the bee bowl survey involved 3 different colors of bowls, small bowls, 3 ounces. And the 3 colors were white, yellow, and blue. This is a protocol used for bee monitoring by Sam Droege. And the bowls were laid out in this pattern in the middle of that Powerline right-of-way. So in between, not in any one of the research plots, but in the middle, just to look at additional bee diversity in the site. And we collected 36 bee specimens in that bowl survey of 19 species, and there was just one sample. And in that same survey, we collected 49 non-bee specimens of 27 morphospecies. And all of this has been recently published in PLOS One. So if you want to read more details about the study or our findings, you can read that paper. So more details about the bees, because they ended up being our primary focus. In terms of bee abundance, the community of bees was actually very evenly divided among the different bee species. So the greatest abundance was Bombus impatiens, but it was only 14.5% of the collection. So it's a pretty even community. Our next most abundant species was Apis mellifera, and then after that, Serotina dupla. And all of the other bee species individually represented less than 5% of the sample. So there were 126 bee species, most of them were pretty evenly distributed. And I just wanted to share this tree map of the different bee genera, so you can compare the relative abundance of the different genera of bees. So obviously, Bombus and Andrena, Lasioglossom, Serotina are all very abundant bees, unsurprisingly. But then you can see that there are less common genera over here on the right, and too many to list out in the box. Overall, we found 28.8% of the bee species of Pennsylvania, thanks to the survey that was published last year. So we could compare our bee diversity in the Powerline right-of-way to total bee diversity of the state. And I think this is a good indicator of how diverse these Powerline rights-of-way can be, given that we only sampled for 2 years and we only sampled those 6 small plots. So just to go over some of our results, looking at the vegetation management scheme and how that affected the pollinators. In terms of bee abundance, we found that there was a significantly lower bee abundance in the low volume basal treatment. And you might notice that the hand cutting treatment is not on this graph. And the reason for that is that the hand cutting treatment was almost impossible to survey properly. And I'll talk a little bit more about that later. But just for the purposes of statistical analysis, it wasn't fair to compare the hand cutting treatment to the other treatments. And so I just included the liters per hectare and the herbicides used in that treatment if you wanted to compare that. But it was lower of bee abundance than the other 2 herbicide treatments. When we look at the continuous effect of liters per hectare on the different bee abundance and bee species richness, we found that the continuous effect was greater than the categorical effect, and that there was a negative effect of increasing liters per hectare herbicide use on bee species richness. Included on this graph, you can also see these triangles. Those represent the plant species richness in the plots when we surveyed them in 2016. And that's just for comparison to see whether plant species richness is also affected by these herbicide treatments. So again, the white boxes are low volume foliar treatment, the gray is low volume basal, and the black are the high volume foliar, which had the highest herbicide application in terms of liters per hectare. We can also look at different measures of bee diversity. So on this graph, panel A is species richness, panel B is Shannon diversity, and panel C is the Simpson diversity index. And this is calculated with rarefaction analysis that allows us to extrapolate the projected diversity index based on our sampling effort. So the blue line is the low volume foliar that tended to have the highest diversity in all 3 different measures. And the green is the low volume basal, which tended to have the lowest diversity, but it was not significantly lower in terms of species richness, just in terms of Shannon diversity and Simpson diversity, which probably means it had a lower species evenness as opposed to species richness. We also used an ordination analysis to compare the different treatments and the different times of sampling on the composition of the bee community. So in this, the different colors are different months of the year that we sampled, and the the different figure types are the different treatments. And when we ran this ordination analysis, the effective month was greater than the effect of the herbicide treatment. So it seemed like treatment was less of a significant factor in determining the composition of the bees. So some challenges associated with the study. Number one, the hand cutting plot was nearly impossible to sample. Partly that was because of brambles, and we were using hand nets, and the hand nets would get caught in the brambles, and that made it very difficult to sample those sufficiently. So in terms of sampling effort, we didn't feel like it was fair to compare the hand cutting treatment to the other treatments. We did provide the data in that publication if you're interested in looking at that anyway. But as a spoiler, the diversity and the abundance were incredibly low, but we didn't feel like that was a fair representation of the treatment. The vegetation management did not follow the original plan laid out, and so our replication and the different categorical effects were slightly different than we had originally planned for this experiment. And the main effect of that was it made the statistics more difficult to run ultimately. We might have replicated things differently if we had known that the vegetation management would be different. Then the third challenge, and probably the big challenge for any one of these surveys, is the taxonomy is very time consuming and difficult, especially when you're including all insects and not just focusing on bees. So one of the reasons we focused on bees in the second year was that taxonomically it's a more tractable group than looking at all flower visiting insects, including beetles and flies and wasps and everything like that. So the takeaway messages are that there was no consistent negative effect of the herbicide on bee abundance. One of the treatments was lower than the others, but it wasn't consistent. But we did see a significant negative effect of the increasing liters per hectare herbicide application on bee species richness. We also saw a negative correlation between plant species richness and herbicide application in terms of liters per hectare. And finally, we found 126 bee species and 179 non-bee morphospecies and representatives of all 6 bee families of North America, including 2 new state records for Pennsylvania. And I'm just highlighting this because, a very small sampling effort, we saw an incredible diversity, which suggests that these rights of way could be really important for pollinator conservation. And I can take any questions. Or I don't know if we have time right now for questions.
Yeah, thank you, Laura. That was a really great presentation, and some of those conclusions are really exciting. We are going to direct all questions for Laura to be put into the group through the chat so that we can move on to our next presentation. But I also want to remind anybody who called in via phone, if you could chat Claudia to let her know who you are. She's working to figure out who is going to what breakout room, and she can't assign people to breakout rooms with just their phone number. So a quick housekeeping reminder for that as well. All right, our next presenter is going to be Emilie, Dr. Emilie Snell-Rood, who is going to be speaking on roadside plants for pollinators and implications for management of roadside habitat. Emilie, the ball is yours.
Great. Thank you so much. And thank you for the opportunity to present some of the work that we've been doing in my lab. And let's see, does this work? Oh, there we go. Okay, great. So before I get started, I want to first acknowledge the co-authors and collaborators on this work, a number of grad students, postdocs, and faculty at the University of Minnesota, and our various funding sources. So over the last several decades, there's been increasing attention on potentially using roadsides as habitat for native plants and wildlife. And this is beneficial not only to provide potentially huge acreage as habitat, but for water filtration and benefits to motorists passing by. Specifically with respect to insects, beneficial insects and insect pollinators, just as an example about the potential of roadside habitat, in Minnesota alone, the average roadside can contain 2,000 to 3,000 stems of milkweed per mile, potentially creating huge habitat in terms of host plants for this declining and near threatened species. And with respect to rusty-patched bumblebees, which are endangered, roadsides, 4 to 5% of roadside sites support rusty-patched bumblebee populations. So there's huge potential in restoring roadsides with native vegetation in terms of supporting beneficial insects and insect pollinators. But there are potential costs as well, because the nutrition of plants along roadsides is kind of complicated with respect to insects. So on one hand, roads accumulate nutrients that are historically limited in availability in most insect diets. So for example, roads accumulate nitrogen from roadside exhaust, and at least in northern climates, sodium from road salt application. And both of these nutrients have been suggested to attract both insects and herbivores in general to roadsides, because they traditionally are limited in availability, but important components of their diet. But roadsides can also accumulate these elements and other things to potentially toxic degrees. So high levels of sodium can become toxic, and then roadsides also accumulate other things like metals, zinc, nickel, cadmium from car general wear and tear, and also contain residual lead from the era of leaded gasoline. Roadsides also can potentially contain pesticides, insecticides spilled over from adjacent agricultural habitat. So the question is, to what extent does roadside habitat present a great opportunity for conservation, or are we just creating a toxic trap that is drawing pollinators in and then killing them because it's full of lead and other metals? So in our research over the last 4 to 5 years, we've been asking this question by first thinking about, well, how do toxins move from the road into plants used by monarchs and other butterflies and also bees? And then at what point do things start to get toxic for these insects feeding on these roadside plants? And then what does this all mean for management? And all of this work is funded through Minnesota State, the Environment and Natural Resources Trust Fund. So I'm going to start here and thinking about how toxins are moving from the road into plants used by monarchs and bees. And I'm going to talk about 2 things and summarize a bunch of work. First, how much do road traits matter, and how much do landscape traits matter? Because if we can distill it down to this, we can potentially then extrapolate to thinking about implications for management. So we'll start with road traits. And for this work, our general approach is to first go out in the field and do surveys of roadsides across. We're focused in Minnesota. Depending on the year, we've been looking at 50 to 100 roadside sites spread around the state, going to the site, collecting plants, nectar, pollen, collecting pollen off of bee legs, collecting caterpillars, collecting bees, and then grinding them up and seeing what's in them to get an idea of how things look out in the field. And then combining this with manipulations, experimental manipulations of the plants, where we plant these native plants in field arrays in the field or in the greenhouse, and then water them with specific amounts of sodium or metals or nitrogen, and look at how that gets into the leaves and the nectar and the pollen. So what do we see? I'm not going to review everything we found, but just as sort of an example of the types of things that we look at, I'll show you the data from the field with respect to sodium. So we see a signature of sodium moving from the road, from winter road salt application, into the soil, into plants growing in that soil. So with respect to milkweed, each point here represents leaves from an individual milkweed plant. Over here, and milkweed's soil closer to the road has higher sodium content, and leaves of milkweed growing in that soil closer to the road also has higher sodium content. You also see a signature of traffic volume, which tends to scale with the amount of road salt applied. So soil from higher traffic volume roads contains higher levels of sodium, and similarly milkweed leaves growing in that soil have higher levels of sodium. So sodium is moving from the road to the soil, to the milkweeds growing in that soil, and then in turn to the caterpillars feeding on those leaves. So caterpillars feeding on milkweed leaves with higher sodium content have higher body sodium content. And we see similar patterns with not sodium, but certain heavy metals, especially zinc, accumulating in roadside soils and getting into roadside plants. So those are just data with respect to milkweed. What about other plant species used by pollinators? We've done a lot of work with common milkweed, but we've also done work in the field with a number of other, mostly native, but some non-native plants. And this is, wrong way. This graph is showing leaves for these plants collected along roadsides with lead as an example, contrasting low and high traffic volume sites. And there's some variation across species, but in general, leaves of species collected along high traffic roads contain higher metal content than those along low traffic roads. So we see patterns across different species of plants. We also see patterns across different metals. So we see a traffic signature for a number of different metals, not just lead and zinc. And we also see a signature of distance from road, but traffic is more important. So we combine these field observations with data from experiments to look at actual uptake of these metals. So just as an example, these are data from a field experiment where we watered with zinc, and all of these different species, if you water them with zinc, tend to take up that zinc and put it in their leaves, and those have greater zinc content in the leaves. Of course, the roadside plants also have additional metals on the leaves in the form of dust, but we can kind of tease this apart. There's also some interesting variation across species where we have some species of plants that take up lots of zinc, and they also take up lots of sodium. So maybe we might want to avoid those species in our plantings. So how much do road traits matter? A fair amount. We can describe a bit about the nutrition of a plant and the toxins in a plant based on the traffic volume of the road and how far away from the road that plant is. So how about landscape traits? So here we've been focusing on two landscape traits. The first I'll mention is adjacent railroad. And adjacent railroad was of interest to us because some previous work has shown that railroads can be associated with heavy metal runoff. And at least in Minnesota, there are quite a number of restored roadsides that also have adjacent railroad. And so we included this in one of our experiments. And interestingly, we see absolutely no negative effect of adjacent railroad, which is good news. And indeed, for some plants, you actually see a positive effect of adjacent railroad. The other landscape feature we were interested in was adjacent agriculture. The idea here being that there might be insecticide spillover into this pollinator habitat. And so here we collected a bunch of milkweeds and did a pesticide screen on them. And almost 70% of milkweed, randomly collected roadside milkweeds, have at least one detected chemical residue. 36% containing insecticides specifically, picked up a lot of fungicides and insecticides as well. Most of the insecticides we detected were this one here, Chloropyrophos. But interestingly, whether or not a plant had insecticides was not predicted by adjacent cropland. So this just shows the probability that a randomly collected milkweed contains at least one insecticide, and there's no difference, yes or no, between sites with adjacent agriculture and those without. So, and we're continuing this work, so this is a preliminary analysis. So while road traits matter a lot for predicting toxins in these plants being used by beneficial insects, landscape traits seem to matter not as much, at least the ones that we were looking at. So given these observations about the plants, when do things start to get toxic for the insects feeding on them? And here I'm going to touch on some of our work on metals, work from the literature on insecticides, and some of our work on sodium. For all of this work, our general approach is to raise bugs in the lab or in the field and manipulate toxins. So we're doing both rearing of bumblebees, bombus impatiens in the lab, and rearing butterflies in both the lab and outdoor enclosures, focusing on monarchs, but also cabbage white butterflies, which are kind of like the lab rat of the butterfly world. Today I'm just presenting the butterfly work, but we do have bee work in progress. And I'm just going to show you data from one of the 4 sets of studies we've done on different metals with respect to zinc, because it highlights 2 take-homes. The first is that toxicity of many of these toxins varies with the species that you look at, and monarchs tend to be more sensitive than some of the other butterflies. So these are data for monarchs raised on a bunch of different zinc diets of increasing amounts of zinc. And you see at some point their survival starts to decline, but they're kind of unfazed in terms of development time. In contrast, cabbage white butterflies, when reared on increasing zinc by small amounts, actually have increases in survival, which we call a hormetic effect, with a slight cost in terms of development time. So monarchs tend to be slightly more sensitive than cabbage whites, which are fairly unfazed by a bunch of different toxins. And then the other main take home is that toxicity that we see in the lab, the levels that start to be toxic are generally much lower than what we're seeing in the field along most roadsides. So these are histograms just showing the amount of zinc in a bunch of different plants. Sorry, this is so small, milkweeds are right here. The maximum being 70 parts per million that we saw at any of our sites. But where we start to see zinc toxicity is between 200 and 300 parts per million. And this highlights a more general take home that if you look in the literature about, okay, if you look across butterflies and moths, what levels of heavy metals tend to be toxic, you get something that looks kind of like this. So things like cadmium or lead start to be toxic around 200 or 300 parts per million, whereas metals like zinc or copper more around 500 to 800 parts per million across a bunch of different species. But what we're seeing in the field is highlighted by these red dots here. The maximum observed in all of our sites is well below levels that tend to be toxic, which is good news. So for overall, how toxic are the metal levels? There's metals all over the place in these plants, but they don't seem to be that worrisome, which is good. What about insecticides? So the most commonly detected insecticide in this work was Chloropyrophos. There's no toxicity data for this insecticide for monarchs. So we've taken some data for other leps, for moths, and we combine that with estimates about how much the caterpillars are feeding on, and get a rough, very rough guesstimate that 5 to 10% of the roadside milkweeds have residues of this insecticide that would kill a monarch, which is kind of disturbing. If this insecticide sounds familiar, it's because it has been in the news because there have been efforts to ban it, and hopefully that will happen again because it is also toxic to vertebrates. So the insecticides, at least some of them that we're detecting, do seem to be toxic and potentially worrisome. So finally, I want to touch on some of the work we've been doing on sodium from road salt. And we started this work a long time ago, looking at the idea that sodium, while it's toxic at high levels, can sometimes be beneficial at low levels because it's an important nutrient that's often limited in availability. So for both monarchs and cabbage whites, small, moderate increases in sodium are beneficial in terms of the development of neural tissue and also muscle tissue, although the way this plays out depends on the sex that you're looking at. But once you get to large amounts of salt, it starts to be toxic. And so in this previous study, the moderate increases correspond roughly to amounts of salt on low traffic roads, and this sort of toxic level is more typical of high traffic roads. So since this earlier study, we've been following up with a number of other lab and field studies to isolate specifically the effects of sodium and look at a number of other traits, and to also tease apart, okay, when does it really start to become toxic? So just as an example, this is a study where we watered plants just with sodium and then fed them to caterpillars. And here you see moderate increases in sodium in the leaves corresponding to a slight dip in survival, significant but slight dip in survival. This was done in the lab, and we followed this up with work in the field to test, okay, well, how does this actually, given all of these data with the lab and all of these different traits, how does this actually translate into performance and fitness of these butterflies in the wild? And so in this study, what we did was we raised a bunch of monarchs and a bunch of different salt treatments, tagged them as part of the Monarch Watch tagging program, and released them, and asked how many actually got to Mexico, and took a bunch of other fitness traits as well. And interestingly, in this study, we were raising outside to induce migratory status. The previous lab results have paralleled so far what we see in the field, and that these moderate increases in sodium characteristic of moderate traffic roads cause a slight dip in survival, but it's not significant, whereas these larger increases characteristic of high traffic roads are more worrisome. And the reductions in survival almost exactly parallel the lab results. And we're still getting tag data back. There have been some delays due to COVID, but you see similar here where the controls do best. The moderate traffic road butterflies do okay, and the highest traffic ones don't do as well. The good news is that these really high levels of sodium in plants are in a very small fraction of milkweed, at least if you look at the histogram of traffic roads in Minnesota. So the majority of milkweeds contain tolerable to beneficial levels of sodium in their leaves. So what does this all mean for roadside habitat for pollinators? I kind of did a whirlwind of data here. Overall, it's remarkably promising with respect to the nutrition of these plants for monarchs, and likely also for bees as well, although we're still working on that piece of it. What does it mean for recommendations? So we can come up with some specific recommendations here. In particular, if we have limited funds for restoring roadsides, we should be prioritizing restoration along low to moderate traffic roads, so less than 20,000 cars a day. At least those are the data that we're drawing from in Minnesota. There's some evidence that getting rid of the plants just next to the road is actually beneficial because you're eliminating the most toxic plants and preventing insects from using the plants that have accumulated the most toxins right next to the road. Planting a diversity of plants, especially some of these species that accumulate fewer toxins, could be beneficial. Continue efforts to ban the most commonly detected negative insecticide, and then continue research on open questions, which we think in particular have to do with the risks and costs of collisions, and also methods for cost-effective restoration. And so here I'll mention some of our current work has to do with this latter area where we're working with the Department of Transportation, getting plans for what was done on roadsides, what seed mixes were planted when and how things were restored, and going back to these sites this summer, collecting data on what plants are there, what pollinators are there, and using that to figure out what is the best way to invest in seed mixes and amount of seeds planted to get the best bang for your buck in terms of what pollinators are there. So finally, I think I'm right on time. I will thank you for your attention, and I know there's no time for questions, but I'll look at them in the chat. So thank you very much.
Thank you, Emily. That was a very interesting talk. I have seen some questions for you in the chat, so please keep those coming. Both Emily and Laura will continue to be looking at the chat, as well as Ashley when she finishes her presentation. So please, if you have questions, please ask. All right, next up we have Dr. Ashley Bennett with the Electric Power Research Institute, who is going to speak on how the utility industry is using integrated vegetation management to support pollinators on utility rights-of-way.
Thank you. I have lost the controls at the bottom of my screen, and I'm not sure how to turn on my camera. Oh, start video. Here we go. Sorry. Thank you, Claire. So yes, today I'm going to be talking to you guys about how the utility industry is using IVM to support pollinators on rights-of-way. I'm going to be giving you an overview of 3 different projects that EPRI is working on in 3 different states: Tennessee, Arizona, and Alabama. Okay. So the very first project I want to tell you guys about is a collaborative project with the Southern Company, where I'm working with Claire Ike, and also Auburn University, where we're working with Dr. Jeff Williams and Dr. Josh Campbell. And they're both on, so if you have questions while I'm talking about this, shoot a chat and I'm sure they can answer it for you. So as I mentioned, we're doing this project in the state of Alabama, where we have 4 different rights-of-way where we're going out and sampling both vegetation and pollinators. The blue dots are representing the 4 rights-of-way under evaluation, and the red dot is Auburn University, which again, Auburn is doing all the field work and quantifying the plants and pollinators in this study. So the objectives for this study were to determine the impact of different IVM treatments on both the plant and pollinator community, plant abundance and richness on rights-of-way. We were interested in tracking the cost of vegetation management practices. And then finally, we were hoping to be able to develop the best management practices that the Southern companies could then implement on their rights-of-way. The experimental design for this study was a randomized block design. If you look down here in the lower right-hand corner, you'll see a right-of-way where we have 3 different triangles. Those highlight the 3 different IVM treatments that are under evaluation in this study. This section is a 1-mile section of right-of-way, and we also selected these sites based on similar plant communities both on and off the right-of-way, and you'll notice off the right-of-way is forested habitat. The 3 different IVM treatments that we're testing in this study include a high-volume broadcast, broad-spectrum herbicide. So if you look in the upper right, you can see the effects of that treatment, which are pretty uniform across that treatment plot. And we also had 2 other IVM treatments that were low volume broadcasts, which is where we used a backpack sprayer to go out and spot spray trees. So you'll see in the picture to the left, individual pine trees that have been targeted, again, with a broad spectrum herbicide. And then the third treatment was, again, going out and doing a targeted application, but using a grass-friendly herbicide. So if you'll notice in this picture, we have a ring of dead vegetation. So that was treatment 2. Treatment 3 was supposed to be not have any effects on the grass. This illustrates where we actually did the sampling on the right-of-way. So both the pollinators and vegetation were sampled in the center of the right-of-way, at the edge of the right-of-way, and then in the adjacent off right-of-way forest. And we sampled pollinators using pan traps, vane traps, and then we also went out and did quadrat sampling for the vegetation. So I'm going to go over some preliminary results from both 2019 and 2020. In each of these graphs at the bottom, you'll see the different treatments, treatments 1, 2, and 3. Treatment 1 was the high volume, broad spectrum herbicide. Treatments 2 and 3 were the more targeted applications. You'll also see in the graphs a purple arrow, and that indicates where the different IVM treatments or when they were actually applied. So we have some pre-treatment data and then post-treatment for both 2019 and 2020. So you'll notice after those IVM treatments were applied, we see a very significant decline in percent grass cover for treatment 1, and then less of effect on percent cover for treatments 2 and treatment 3. And we tend to, for the rest of the season in 2019, tend to see less of effect on that grass cover in treatments 2 and 3. When we go and look at the results for 2020, we begin to see that treatment 1 grass cover begins to recover a little bit by 2020. But in general, later in the season, we still tend to see higher percent grass cover in treatments 2 and 3. It's not always significant, but it tends to be higher. When we look at the results of forb cover for each of these treatments, we see a similar result. So here again are when the IVM treatments are applied, and we see a significant decline in forb cover for treatment 1, which is again the broad spectrum herbicide application, and less of effect for treatments 2 and 3. And that tends to hold for the rest of 2019, where we're seeing less of an effect on forb cover for treatments 2 and 3. When we go and look at 2020, we do see that treatment 1 is similar to the grass cover. The forbs also are starting to recover in that treatment 1. But in general, especially late in the season, treatment 3, we tend to see higher forb cover. It's not significantly different, but it tends to be a little higher. When we look at the results for pollinators, irrespective of the IVM treatments, we are finding that the right-of-ways are supporting more pollinators than off right-of-way habitat. So you'll see we have about 90% of the bees coming from on the right-of-way and about 10% of the bees coming from the adjacent off right-of-way habitat. When we look at the effects of the IVM treatments on the pollinators, you do see a decline in bee abundance, but we actually don't see any significant differences across those treatments. And I'd like to point out that in 2019, if you look at June 2019 and June 2020, we see a similar decline in 2020. So some of this decline may be due to the IVM treatments, and some of it may just be due naturally to the number of flowers and forbs we have blooming on the right-of-way. Okay, so some key findings from this very first project. We are finding more bees on our right-of-way than the adjacent off right-of-way habitat. Treatment 1 tended to have the largest effect on both grass and forb cover in the first year, but we did tend to see that those grasses and forbs start to recover in that treatment 1 by 2020. The grass-friendly herbicide tended to have less an effect on both grass and forb cover, and it has another benefit of applying less chemical, which can also potentially be a cost savings for the utility. We did not see any significant treatment effects for the bees. We are in our 4th and final year of sampling for this project, so next year we'll be wrapping up this study and hopefully being able to suggest some best management practices for the Southern Company for treatments on their rights of way. So I'm going to transition to the second project I'd like to tell you about, which is another right-of-way study that we're doing in Tennessee in collaboration with Tennessee Valley Authority, the Southeast Grasslands Initiative, and Mississippi State Entomological Museum. So for this project, as I said, we're doing this study in Tennessee and on the Cumberland Plateau. So if you look at this graph, this yellow area is the Cumberland Plateau. And what most people might not realize is most of the southeastern United States at one time used to be covered by quite a bit of prairie and savanna habitat, over about 100 million acres. And the Cumberland Plateau in particular was covered by prairie and savanna habitat. And most of this habitat has now been lost to forest succession. So just here's another picture highlighting where that prairie and savanna habitat is or was in the state of Tennessee. And if you'll pay attention to Chattanooga down here in the center of the graph, in this next photo, you'll be able to see Chattanooga and where some of our study sites are located this year for 2021. So we have 5 rights of way that we're sampling for both plants and pollinators on rights of way in Tennessee. And all the photos in this section of the presentation are photos that have actually been taken of plants and pollinators at our study sites. So the Tennessee Valley Authority wanted to know how compatible their IVM management was with some of their conservation goals. And they also wanted to know what native plants and pollinators were actually being supported on rights of way in Tennessee. So again, for this study, we were going out and sampling plants using transect and quadrat sampling, and going out and sampling pollinators using beetles, netting, and malaise traps. Again, we're sampling vegetation and pollinators both on and off the right-of-way. And here's just an illustration of where we're doing the plant sampling, which are the black dots, and the pollinator sampling, which are the purple stars, both on the right-of-way and off the right-of-way. Tennessee Valley Authority was interested in not only bees, but other pollinators, which is why we're using a variety of sampling techniques from sweep netting to beetles to malaise traps, so we can quantify beetle pollinators, moths and butterflies, as well as flies. So some preliminary results from our 2019 data. So in 2020, we actually weren't able to go out and sample because of COVID. But for 2019, we found that plant richness was about 2.5 times greater on the right-of-way, which are the blue bars, compared to the off right-of-way forested habitat, which are the green bars. Utilities in general are very interested in suppressing woody trees and shrubs on their right-of-way through their integrated vegetation management practices, while encouraging grasses and forbs. So we wanted to be able to compare the ratio of grasses and forbs to woodies on our right-of-way, and we are finding about 6 times more grasses and forbs, which again are the green bars, compared to woody vegetation on the right-of-way. So that's suggesting those IVM practices are really helping suppress woodies, promote grasses and forbs. Well, in the forested plots, it was about a 1-to-1 ratio. I did want to highlight some exciting results. We are finding a lot of rare plants on our right-of-ways in Tennessee, both plants that like wetter conditions, like these orchid species. So one right-of-way in particular, we documented 7 different orchid species. We're also documenting a lot of native grasses and forbs, prairie grasses and forbs on our right-of-way. So for the pollinator results, very similar to the study in Alabama, we're finding much higher abundances and diversity of pollinators on our right-of-way. So 2 different sampling techniques, bowls and netting, we found 16 times and 14 times more bees on the right-of-way than in the adjacent forested habitat. And butterflies, similarly, 4 times higher abundance and richness on the right-of-way. We did actually document 1 vulnerable species, the Leonard skipper, on a right-of-way in Tennessee. Okay, so I'm going to tell you guys about one more right-of-way project that we're actually just getting started this year in Arizona. And for this project, I'm collaborating with the Salt River Project, which is a utility in Arizona, Northern Arizona University, and the USGS. So for this study, again, I'm taking you to Arizona, and our study plots for this project are just outside of the Phoenix area. And for this study, our goals were, again, to determine the value of IVM practices on rights of way to native plants and pollinators in 3 different ecoregions in Arizona. We also wanted to compare different IVM treatments for their ability to suppress woody vegetation while encouraging grasses and forbs and hopefully having a positive impact on pollinators. So as I mentioned, in this study, we have a right-of-way that crosses 3 different plant communities in Arizona. So we're going out and sampling pollinators in the Sonoran Desert, which is the lowest elevation, the pinyon juniper plant community, which is the mid elevation, and the ponderosa pine, which is the highest elevation. Here's just a map showing you where our Sonoran Desert study sites are, our ponderosa pine, which are the green, and then our pinyon juniper sites, which are purple. Again, in this study, we are sampling pollinators and vegetation both on and off the right-of-way. We are also conducting IVM treatments in the ponderosa pine plant community. In some regions on this right-of-way, SRP is only able to mow, and this mowing can cause increased growth of some woody plants. So SRP was interested in whether an integrated approach using both targeted herbicide applications would actually provide better suppression of woody plants on the right-of-way while encouraging grasses and forbs. So the 4 IVM treatments that we're going to be testing in this study are a control, an herbicide-only treatment, a mechanical-only treatment, and then a combination of mechanical and chemical applications for the final treatment. Similar to our previous 2 studies, we're going out and sampling the vegetation, and we're doing this using quadrat sampling where we're identifying all the plants and species. And we're also sampling pollinators both on and off the right-of-way using netting along transects and also doing timed quadrat counts. The Sonoran Desert, we have 7 study sites that we're sampling. The pinyon juniper as another 7 study sites. And then the ponderosa pine, we have additional study sites here because we're sampling both on and off the right-of-way in addition to those IVM treatments. So we are just, as I mentioned earlier, starting this research this summer. So I only have some very preliminary data to share with you from the Sonoran Desert. Someone's drawing on my slides, that's okay. So we found 48 different species of flowering plants in the Sonoran Desert, and we have found similar abundances and richness both on and off our right-of-way. So you go down and look at this table. If you look at plant abundance, we found about 52% abundance on the right-of-way and 48% off, which is very similar. The richness for plants was slightly higher at about 55% compared to 45% for plant richness. Here's just a common list of the flowering plants that were documented in the Sonoran Desert right-of-way. And here's just a photo of what some of those most common plants look like. I'm actually not that familiar with plants in the Sonoran Desert, so really the only ones I recognize were fleabane and creosote bush. So the next steps for this project is we're going to continue our pollinator sampling in the pinyon juniper and ponderosa pine plant communities this summer. We plan on implementing our IVM treatments this fall of 2021. Then we're going to be continuing our plant and pollinator surveys in 2022 and 2023. And I think that is the end of the data I wanted to share with you for our 3 different project areas. And I think I'm right on time. So let's see. We're going to, I've been seeing a lot of questions going on in the chat, so it seems like we've been getting a lot of good chat, but this kind of brings us to the end of our 3 research talks highlighting right-of-way and pollinator research.